Last but not least, it's a great pleasure for me to introduce Yannick Wurm as our final speaker, keynote speaker. So I met Yannick virtually through Bio.js, actually. I remember it was because of the Google Summer of Code that you asked us if we had an internship for the Google Summer of Code. I don't remember what happened in the end. Maybe it wasn't chosen or...
Nothing happened, but I don't remember the details.
And then, his group has developed BioNode, which is the application of Node.js into a kind of biological purpose. BioNode has become one of our component type of Bio.js. Sorry? Probably I'm saying I'm bullshitting here, but never mind. So Yannick does a lot of genomics with ants. He's looked at their behavior, their social interactions, I understand. One time I called him, where were you? You had been in Borneo looking at ants and these kind of things. So it's a kind of very interesting type of research. So so I would like to introduce you to Yannick. Thank you, Yannick, for accepting being our keynote speaker.
Thanks very much, Manny, for the invitation. And thanks to all of you guys for hanging around. I'll try to be relatively quick so we can go have some drinks. I'm going to start by my thank you slide so that we have more flexibility in the timing of when I finish. There's some people in my lab. I've been at Queen Mary University in London for 3.5 years now. I'm going to talk about some of the work that they've been doing. Previously, I was in Switzerland, and there I was in the lab of Laurent Keller, who was at University of Lausanne, but interacted very closely, and I was also a member of the Swiss Institute of Bioinformatics as part of Yannis Xenarios' group. We have a little bit of funding. If anyone has more funding they want to throw at us, please get in touch. We always like money. So when I tell people that I work on ants, usually they ask me, okay, the red ones or the black ones? But I'd like to, fix people. There's 20,000 species of ants. There's green ones, there's orange ones, there's spiky ones, there's hairy ones, there's shiny ones, there's some with big heads, there's some that look like weapons, there's some that look like aliens even. There's a huge amount of diversity. These are all workers. It turns out that what these 20 species of ants have in common is that they live in organized societies. And this type of organized society independently evolved also in the termites, the bees, and the wasps, whereby a few individuals reproduce, and then the large majority of individuals are sterile workers that tend the nest, feed the brood, etc. There is as much diversity in the social lifestyles of these ants as there is in the colors and morphologies of the workers I showed you a minute ago. And because it's already 5:38, we're going to have a bit of a YouTube break. So I'm going to show you some of the diversity of lifestyles. Go into basically in the Americas, the rainforest, there's lots of trees. These leafcutter ants, they cut the leaves and they bring them home to the nest. Now, the trees don't want to be eaten, and so, like nicotine and other plants, they put poison into the leaves. And then anyway, cellulose is a bit challenging to digest. So what these ants do is they grow a fungus on these leaves. So this white stuff is a fungus. They're cultivating this fungus. They started cultivating this fungus 50 million years ago. They invented agriculture long before we did. And so the fungus digests the leaf, and the ants eat the fungus. The colonies are huge. So here, a single colony died. Some people poured some cement inside and then waited for it to dry and dug it out. This is a single colony, single queen. I don't know how many million workers. You can see the tunnels. You can also see these balls. Each one of these is a separate fungus rearing garden. And they're actually pretty cool because they have separate vents, kind of one for old air to be evacuated up and then fresh air to come in, because otherwise things don't work. Really amazing stuff. Another way some ants work with leaves is in the weaver ants. And so these are more Southeast Asia, Australia. You might have seen them. So they're up in the trees. They take leaves and they pull them together. And once 2 leaves are pulled together, the workers take the larvae and use the silk made by the larvae to stick the ants together, to stick the leaves together. And so, a lot of insects, they build a cocoon to protect themselves during metamorphosis. That doesn't happen here anymore. The silk is only used to stick the leaves together. Here they're still working on it. And then you end up with these aerial chambers that are closed, that are protected, and the colony, lives in a set of these with the queen, the brood, the food storage they have. Another funny home is in this Brazilian ant. These guys live in the sand. That's the entrance of their nest. It's open during the day, but every evening they close the entrance. And it turns out they do that from the inside, but also from the outside. And so to do that, every evening, a handful of workers, maybe 5, 6 workers, stay outside. There's the entrance of the nest. Here's after they've started closing it. And then here's kind of the half hour around dusk accelerated. So the 5 workers that stayed outside close the entrance and then wipe everything clean. And by the time they're done, I wouldn't be able to tell you, where the entrance of the nest was. These workers that stayed outside, they then head off into different directions, and they die during the night. And so it's a rare example, I think, to my knowledge, it's the only one outside of humans, of preventive self-sacrifice. So we interpret that they're, hiding the nest entrance to protect it from intruders. The most likely intruders are actually other ants. Ants with the driver ant, these guys are in Africa, or the army ant lifestyle. Here, the colony is several million individuals strong. They need so much resources that they don't have a colony in a single physical place. They're kind of always on the move, carrying with them their, here we see them carrying their brood. Point is, there's a huge diversity of lifestyles in the ants. And the fact that they live in societies, plus the diversity of lifestyles, makes social insects wildly successful. If you take a chunk of rainforest and you count all the animals and you weigh them, social insects make up almost 1/3 of the animal biomass. Mammals are pathetic in contrast. Because of this, they've been very well studied. We know quite a bit about their behavior, their morphologies. All phylogenetic trees, all the systematics was based on morphology until recently. We know a lot about the contexts in which social behavior can evolve, the types of conflicts that can occur in societies, quite a bit about their ecology, but very little about the genes underlying their behaviors. And so to talk a little bit about that, we're going to talk about another ant. And this ant is commonly found in America. Are there any Americans in the room? One. Sorry. I love America too. Happy 4th of July tomorrow. When the rest of us go to America, we meet this immigration guy. And, he asks us, why are you coming to America? How long are you staying in America? Are you a terrorist? And all kinds of other questions. And usually when I go to America, it's to study fire ants. And, I tell them I study fire ants. Systematically, I get a really emotional response. Fire ants, they're nasty critters. They attacked my dog. And then the guy goes on and on about his mother-in-law, and they had to move house, and every time. Not the same story, huh? But an emotional reaction. The reason is that they're a huge problem. So in the 1930s, fire ants from South America made it through immigration to Mobile, Alabama. People were importing plants, and these ants were in the soil. And since then, they've spread to warm parts of the southern US, but also to many other parts of the world. They're highly invasive. They have very high colony densities. They make big mounds, so it can even damage farm machinery. But most people know them because they have a very potent sting. They latch onto you with their mandibles, and then they inject their venom just like a wasp would. It hurts, hence the name. It burns for about a month, hence the name fire ants. I am happy to say that the photo you're about to see is not me. This person might have fallen on a fire ant mound. I don't know. That must hurt. Because of that, the Americans have invested billions in studying these ants. They are the best studied ant. They're the one where we know the most about the natural history, about the colony life cycle, everything. So a lot of people are interested in eradicating them. They're not here. They don't bug me that much. I'm interested in something else in them. I showed you there was a bunch of different lifestyles in ants. Well, this species is special because here within a single species, there's two different types of lifestyles. So there's some colonies that have strictly one queen and other colonies that have multiple queens, up to 100 queens. And in addition to the colony number, there's a few other differences with regards to territoriality, aggressiveness, etc. As a side note, maybe to you and me, having one or many queens doesn't seem like much. To an ant, it is a lot. It's a really fundamental aspect of how the society is structured.
Definitely the same species?
Absolutely 100% the same species, yeah. But there's these two social forms. And back in the 1990s, these two guys, Ken Ross at the University of Georgia, Athens, and Laurent Keller, who was in Switzerland, they got some money to do population genetics on them. And this was before full genome sequencing and even HapMap and even microsats and even RFLPs. They used something called allozymes. Now, if anyone here remembers allozymes, don't worry, we won't think badly of you. The concept is really simple. You take some ants, you crush them up, and you load the resulting mulch onto a gel. Here that was done for 3 samples, and then you apply a current. And that current separates the proteins making up the ant according to the charge and the mass of those proteins. And then here, for example, at this locus, there's 2 variants, a faster, I guess a smaller variant, and a bigger one. So homozygote, homozygote, heterozygote at that locus. This was back in the 90s. They didn't have millions of SNPs. What they had was 14 such markers. And what they saw confirmed that these two social forms, it is a single species. At 13 out of the 14 markers, there was genetic variation as you would expect when comparing random individuals. But one of the markers stood out, GP9, general protein 9. In that one of the alleles existed only in multiple queen colonies. So for this one locus, GP9, the major allele, well, every single queen colony only had the major allele, and the minor allele could only be found in multiple queen colonies. And that was the first time some sort of a Mendelian element was discovered to be associated to how a society is organized. And now over the next few seconds, I'm going to summarize kind of the 20 following years of behavioral work. I apologize, it's a bit complicated. It's a special system, but so please try to pay attention. So there's 2 possible alleles, and to make things even more simple, they named one of the alleles Big B and the other one Little b. Sorry about that, it was before my time. Little b individuals, they die when they're very young. So that makes the system a little bit easier. In a single queen colony, everyone is Big B, Big B. In a multiple queen colony, the queens are heterozygotes, and the workers, they're kind of 50-50. Some are homo, some are heterozygotes. If you add another queen to a single queen colony, she's executed by the workers. If you do the same thing to in a multiple queen colony, well, a Big B queen will also be executed. But a heterozygous queen smells different. There's a different odor on her cuticle, on her skin, and she'll be accepted into the colony. This has been described as a selfish or a green beard gene system, because here the workers that have the little b allele, they execute reproductive individuals that do not have the little b allele. So effectively, the workers with the little b allele are helping the little b allele reproduce, even though they cannot reproduce themselves.
So it's not the red workers that do the execution.
90% correct. Yeah, there's a little bit more information is needed. But red workers here would execute anyone. Red workers here, they're accepting these guys. How much red workers contribute to executing red queens here is unclear.
But you would expect red workers to kill the blue queen, like on the left, but they don't, if there are blue guys among them.
So here, red workers will kill blue queens.
There they go.
Here they don't. So, and you can manipulate this actually. So if you change the proportion of blue workers in the colony, that will end up changing the behavior of the red workers and make them accept, one versus multiple queens as you.
Can you introduce blue workers into the red colony?
Yeah. So you can do that. It's really easy to do the manipulation because you can keep the whole society in a box like that big, and then they're aggressive to each other as adults, but as brood, as larvae, you can transfer things over. And that makes it really easy to modify this proportion. And so if you have only red workers with no queen, they will kill any blue queen you put in. If you have homozygous, if you start adding blue workers, after a while, they will start accepting blue queens and only blue queens.
Sorry,
Yeah.
I might have missed. Are you talking about the same species?
It's a single species, yeah.
Okay.
There's two types of social organization in this single species. Yeah.
You can say the difference between the queen and the worker at the newspapers.
So in 99% of cases, there is no genetic difference between queens and workers. So whether a female egg so workers are female, queens are female. A female egg is diploid, a diploid egg is female, and it's during development. Maybe second larval stage, third larval stage, depending on environmental factors, how much food, whether there's already a queen in the colony, the quality of the food, maybe the time of the year, which will determine whether an individual is able to determine into a queen or a worker. There's a bunch of interesting questions linked to that. One of which is that the workers live about one season, like 6 months or a year or something like that, whereas the queens, some of the ants that are the black garden ants that are walking around outside, the oldest one we know of, basically somebody caught a queen and kept her in a box until she died. She was 30 years old when she died. And so there's a huge intraspecific variation in lifespan. Genetically, they're identical. Somehow they're using their bodies differently, and one's able to live 60 times longer than the other.
If I could have just a quick tangent. How do you define species by being able to interbreed between individuals?
Here, any definition works.
Oh, yeah.
Yeah, but yeah, that's a good definition. Yeah.
And then, but then does that imply that of the 20,000 ant species, none of them can interbreed?
Correct.
Okay.
Yeah. There's much more. We have, one word for dog. They all look different, but it's all the same species, and those are all wolves. We have one word for ant, and there's 20 to 30,000 species, depending on how you. Yeah.
Please make my
Maybe that's a project for the hackathon tomorrow. You enter any text and ants make it. Yeah. Anything else now? Yeah.
Was there other males in these colonies?
So the males, so if an egg is haploid, then it becomes male. So an unfertilized egg becomes a male, and they only live like 2 weeks or something like that. So once they're adult, they wait until they can go on a mating flight to fertilize a female, and then they're over. The weird thing is that, so the male fertilizes a queen, and the queen, she usually just mates that one time, when she's also just 2 weeks old. But the queen lives up to 30 years, and she has a special organ called a spermatheca, and she keeps the male's sperm alive for those 30 years. And somehow there's enough sperm in there to, keep laying 400 babies a day for 30 years. Anything else? Okay, right. I remember where I was now. Okay, sorry, we're going to have to see the animations again. So when they found this association between GP9 and the social form, and that they kind of demonstrated that this Mendelian locus, this genetic element, was responsible, people, there was quite a bit of discussion. So, were they really lucky? Did they find the master switch, the master gene, master regulator for determining how these societies are organized? Or maybe, they only had 14 markers. So statistically, maybe this gene is a marker for a 14th of the genome. Who knows? Well, nothing happened, for 10 years, 15 years, because there was no way to go further into this system. You guys have, I'm sure, seen this graph many times. 2007, 2008, everything changed for biology. Because a project that would cost $15 million and require 300 collaborators not that long ago, well, now it can be done, the data generation part of that project, sequencing data generation part of that project can be done by a master's student. And so the sequencing technologies allowed us to take this project from the 90s and bring it back into modern day. So to do that, we first sequenced the genome of a big B male. As I said, they're haploids, so that makes things easier. We also sequenced the genome of a little b male. And then we did rad sequencing. So what rad sequencing means is basically we took a queen and we let her lay some eggs. And they develop into males. Some of them are big B, some of them are little b. She lays many males. And then if this represents the haploid genome of this male, take the haploid genome, and then you cut it with EcoR1 digestion enzyme. And then we pull out the 100 nucleotides right next to every EcoR1 site. We label those with a unique identifier, 5 nucleotides that specifically identify this male. And then we take all those little sequences and put them in a tube. We do that, again, for many other males. Some of them are big B, some of them are little b. And each one has a unique 5 nucleotide identifier. And then we send all that away to sequencing, maybe here. Essentially what we're doing, by pulling out the sequence right next to the restriction, the EcoR1 restriction site, we're sequencing the same 0.1% of the genome in all of these individuals. And in some cases, there's some polymorphism. Sometimes, the sequence is the same for everyone, but sometimes there's a single nucleotide polymorphism there. After massaging the data a bit, you end up with a nice table where for each site and for each individual, you get a genotype. That kind of data can be a bit, well, kind of the first way of looking at that kind of big matrix can be to do a principal component analysis, which basically consists in principal component analysis is blind. You give it all your data, and it somehow magically pulls apart the data according to the biggest axes of variation. And so I'm going to show you what we see on principal component 2 versus 3, and then on 1 versus 2. So here, each dot is one male. So the input was the vector of the genotypes at 2,000 loci of that male. That's a different male. No particular spatial structure or anything if you look at principal component 2 or 3. But if you look at principal component 1 here on the x-axis, the data clearly clusters into 2 groups. And it turns out that's fully associated to GP9. That suggested that, basically that told us that a large proportion of the genetic variance there is between individuals is completely associated to the genotype at GP9. We did a bunch more analyses, including building a linkage map. We end up with 16 chromosomes. Each one of these bars is one chromosome. It turns out that across about 2/3 of this last chromosome, chromosome 16, about 2/3 of that chromosome, there's no recombination between the big B haplotype and the little b haplotype. So GP9 gene is somewhere in there. And there's never any recombination going on between GP9 little b and any of the things around it versus the big B haplotype. Having established that this thing, which previously, where they had only one marker, is actually a big chunk of chromosome, we can start thinking about this as a social chromosome because it determines the social structure. And the clear analogy is with sex chromosomes. And once you start thinking about it in this manner, some people would call it a supergene. We called it a social chromosome. Well, X and Y sex chromosomes, they look really different. It's clear why they don't recombine. So why would there be no recombination between kind of the big B version of the social chromosome and the little b version of the social chromosome? It turns out that this is largely due to structural differences. So they weren't easy to find, but I'm going to show you just a small example of a structural difference. Here's a small portion, so just 200,000 base pair. Here is the version on the big B social chromosome, and here's the equivalent, so the version taken from the assembly of the little b male's genome. What's in blue is things that align perfectly. What's in red here also aligns perfectly, but it's flipped around the other way. And so if part of the sequence is flipped around, it's easy to imagine that, well, when meiosis happens, it's hard for the cellular machinery to make things align to be able to do recombination. And so that kind of structural, kind of an inversion, is likely responsible for the absence of recombination. So I've established that there's differences between the big B and the little b version of the social chromosome. You can predict that little b will degrade, and that prediction is based on two things. Basically, population genetic theory makes it clear that if you have a smaller number of individuals in a population or in a species, selection will be less efficient, and you'll have more degradation. And there's a smaller number of little b's than there are big B's in the species, than there are normal chromosomes in the species. The other reason is that little b individuals die. So if a deleterious mutation appears in little b, there's no good way for natural selection to remove that deleterious mutation. And so you can predict that little b will degrade the same way the human Y chromosome has degraded. We found some evidence of that. I showed you this graph earlier. Here, these white bits, they're basically chunks of DNA that exist in little b that don't exist in big B. And typically, they're transposable elements or other repetitive elements like microsatellites or mini-satellites that have expanded in little b compared to big B. That kind of thing is deleterious. Another really easy measure is the quality of de novo genome assembly. If we look at the little b specific part of the de novo genome assembly, the contigs we get are far smaller than anything else, and that's likely due to there being tons of repetitive sequences in little b. When I went into this project, I was hoping to find, I don't know, 5 genes, 10 genes. It turns out there's more than 800 genes in this region. So some challenges are ahead to actually figure out which genes in this region are actually responsible for the differences in behavior between the two social forms. I'm going to skip a bit. So here basically we've determined that a large non-recombining thing, a supergene, a social chromosome, is responsible for this kind of new way of organizing a society or different way of organizing a society. I failed to mention that the ancestral state is single queen, so multiple queenness is a derived state. It turns out that there's a few other species where you also have single and multiple queenness in a single species. The one that has been looked at, it turns out there's also some kind of social chromosome, but it's a different one, completely unrelated chromosome to this one. But nature somehow found a similar genetic architecture to the independent or convergent evolution of multiple queenness over there. And it's likely that this kind of genomic features have involved in a bunch of innovations, and there's evidence to support this also from butterflies, from studies on birds, etc. To summarize this, we have this unique system in the fire ant that has been very well studied, where we've kind of gone at it with modern tools and found that the way societies are organized there is determined by a pair of variants of a social chromosome. Generally, we're quite interested in the interplay between genome evolution and social evolution. I'll spare you the details, but something else that we've started looking at is using some genomics tools to look at pollinator health. Most people looking at pollinator health have been using, behavioral things, experiments that are very long to implement. There's really good opportunities to use population genetics approaches, sequence things out in the wild, and then see, based on analyses of their genomes, how different environmental challenges, be it pesticides, be it climatic changes, are affecting them and what their potential resilience is to future changes. For all of this work, well, we end up having to use a lot of bioinformatic tools, and we end up creating some. And obviously, there's, the computer doesn't care if the ACGTs come from fruit flies or humans or ants. So some of these we've open sourced. In a general sense, I think it's important for everyone to realize that genome bioinformatics is really hard. And I would argue that it's a lot harder than many other data sciences. So you have the general thing that working with large amounts of data is challenging. But furthermore, biology is really complicated, and we don't understand a thing about it. Most of us still think of a genome sequence as a one-dimensional string of ACGTs. In practice, it's wrapped in 3Ds, and there's all kinds of weird stuff going on. Our field is super young, because the data we're dealing with, the price of data generation has gone down 10,000-fold in the past, 7, 8 years, something like that. That's huge. Nothing goes down 10,000-fold in any field except this one. And maybe the biggest challenge is that most of our researchers, our PhD students, our undergrads, our staff scientists, and importantly the decision makers too, don't have any computational training. And so that makes it really hard for them to evaluate the risks involved in this kind of research. Compare that to physicists where you learn quantitative stuff and how to program from the youngest age. Biology undergraduate degrees are slowly adapting to this, but it takes time. As a result of many of these things, I would argue that our tools suck. They're badly written. They won't install. They haven't really been tested. The output, you have 10 tools that you have to test. Okay, 3 of them give you something that's more or less okay. And at the same time, the data sizes keep growing, and then the frickin' data formats keep changing. It's challenging. I think we can summarize the challenges, and this has been done by others today in much more eloquently, that there's challenges with handling data, with analyzing data, and with visualizing data. And these 3 aspects play back and forth off each other. Because to visualize something, well, then you decide, I want to visualize it some different way. And in practice today, that often means, okay, to visualize it slightly differently, I need to spend a week, writing code and debugging why the scheduler isn't working and whatever, just to get a slightly different visualization. It's not an efficient process. I think this community is powerful because most of the people in the room understand the challenges involved in all of this. And so that creates some really great opportunities for us. I'm going to close by showing you some of the tools that we've contributed to. In creating them, we hope that they suck a little bit less. And part of that, so those who are based in the UK, check the software.ac.uk site this is a lobby that tries to convince people at all levels, including funding agencies, to do things better in terms of reproducibility in science. Software being defined as anything that is a line of code that could potentially, that affects the result. First tool is Sequence Server. This started out based on BLAST. Does anyone not know BLAST? Anyone not know BLAST? Okay, there's 2-3 people who don't know BLAST. It's the most commonly used tool in the whole of biology. It's been cited 100,000 times. Like there's 2 or 3 publications, add the citation counts, 100,000. It's also from the 1990s initially. The interface really sucks. So if you go on the NCBI, which is the standard place where a lot of people do BLAST, you have to decide, there's several different algorithms to choose from. You have to choose which algorithm you're going to use before having entered your data, despite the fact that for some combinations of data, you don't need to choose. If you have a protein query and you're going to BLAST against a protein database, you don't need to think. There's only one possible algorithm, and it's a really basic thing. And yet the people making the user interfaces for BLAST, in 20 years of user interfaces, they still force people to choose the algorithm before. Anyhow, there's some challenges with the interface. And then it's also really hard to use on custom data because the big repositories, it can take years before newly generated data is available on there. When we started getting some genetic data for ants, I suddenly became very popular because everyone started asking me to BLAST stuff. Initially, that was flattering, and then rapidly it became annoying. And so we ended up creating a small website, antgenomes.org. There's nothing on it. It's just a BLAST interface. It's not worth checking out. What is worth checking out is the package that we open sourced, and that's Sequence Server. And basically, the philosophy behind Sequence Server is we take BLAST and we want to make it just easier to use. And that means easier to install, easier to use on custom data sets, easier to share those custom data sets, so by setting up a small server. And then the interface for actually using it tries to be a bit smart. Installing it on most Unix systems is relatively easy. Gem install Sequence Server, and then you type Sequence Server. If there's no config file, it asks you, OK, where are your databases? How many threads do you have? If BLAST isn't installed, it downloads the appropriate version of BLAST for your platform. And then if you only have FASTA files that are not yet converted into BLAST databases, it reformats them so that BLAST is happy. And then it uses kind of the internal Ruby web server and launches that on localhost, and you end up with a funky interface. How much for time? OK, should we do this? Oh, I got a message, but I'll keep that one for later. Firefox. OK, so here's the standard interface. You paste in your sequence. Oh, look, it detected it's a protein sequence. That's nice. If you choose a nucleotide database, you can choose additional nucleotide databases, but all possible protein databases are deactivated because BLAST would not be happy if you tried to BLAST both of them. Furthermore, protein query, nucleotide database, there's only one possible BLAST algorithm, so we auto-select that one. Let's say we want to do instead against a protein database. OK, the button changes. We run our query. We end up with a small overview. Here, 2 sequences were blasted. It looks like a standard BLAST report. We try to make it a bit less ugly. And then there's a few ways. If you want to select it, you can. If you want to select several of them and hide them away, then afterwards you can download just those guys. Or you can also use a viewer. This is actually a bio.js component that's very slightly modified. Voila. Check it out if you haven't already. And so this, it's really simple. It's just a wrapper for BLAST. But but it's set up as a permanent server in about 200 labs worldwide, and then many people, just based on Google Analytics clicks back to our documentation pages, suggest that there's many, many users. I'm going to talk about one other piece of software, and that's BioNode. And this slide could be misinterpreted somehow. This is my PhD student, Bruno, who's very sorry he couldn't make it. This was not the intent. Here's the time wasters. In doing this kind of stuff and in doing our analysis, we realized that we were wasting a bunch of time. One of it is when we're making an app, sometimes you develop something that's supposed to be pretty in the web browser, a function in the web browser, and then you realize, oh, we need it on the server side. And then the client is in JavaScript, and then the server that's in Ruby. OK, then you need to recode it. Pain in the butt. Another challenge is things stalling. Pipelines with data coming in from different sources. Sometimes there's a fail. It's hard to detect. Can happen at all levels, when retrieving data from NCBI or the scheduler. And then another thing that came up a few times, sometimes you need to use just a specific library, or you have a specific type of data that you want to analyze. It turns out the only kind of prefabricated thing to analyze it with is a library that's in a different language. And so then you have to use, kind of learn how to program in that language just to access that one library. Based on that, and then the specific needs of Bruno's PhD project, emerged BioNode. And for those who have not yet heard of Node, Node is kind of the way you can do JavaScript on the server. The way you can use JavaScript to do real work, not in the web browser, but on a server, and make it a respectable language. It's wildly successful. So here's kind of the number of BioNode packages has been going way up very recently. So there's far more Node packages than there are now Python and Ruby packages. Here's the website. There's quite a lot of crosstalk with Bio.js and with a project called DAT, which is a versioned data, a manner of storing data in a versioned manner. And I'll first run you through a small aspect of how the project is, recommendations for building modules. So modules should be small. They're built first for working on the server side. But when there's no reason why it shouldn't work on the client side, they should also work on the client side. And then additionally, they're made in a manner, or they should be made in a manner, where you can just use them as a normal command line tool. And I'll give an example of that in a second. And then, because we've been transitioning from a type of working where kind of you spend 3 months generating data and then 6 months analyzing it, that's the past. And then now, increasingly, we're constantly generating data and increasingly have to analyze it in real time through USB sequencers, but through many other things as well. Well, it should do streaming as input and as output, support it at least. Here's an example of what some BioNode code looks like. It's JavaScript. JavaScript's a bit ugly. That's OK. Here we have an NCBI object. We tell the NCBI object, OK, look in the SRA, NCBI's SRA, for anything that matches Solenopsis invicta. That's the fire ant. Whatever you find, do two things with it. One thing is send it to something called fork1, and the other thing is send it to where we're going to store data. Fork1 actually goes to two different places. So fork1 goes to here, where these things happen, and then fork1 also goes to here, where something else happens. And the first thing that happens, and these can happen at the same time, the first thing that happens is that we get metadata about what the biological sample was underlying that set of sequences from the sequence read archive. The second thing that happens is we look if there's any publications linked to that data set, and then pull those out and also end up sending them to the data archive. That last line isn't shown. Point is that you can make complex pipelines, and when different steps happen at the same time, they can happen at the same time, and these streams they can be processing data as they're coming in. However, that's not the only way. Every module that is written can also be used as a standard Unix command line tool. And so here, for example, the other way is I can ask, it for URLs for assembly of Solenopsis invicta, and then that'll just pull out the site where I can download that genome assembly. Every tool can also be used in the command line, and then things can be piped together to do combinations.
It's not easy to do forks. Or is it the ST?
It's a bit creative, I think, to do forks on the command line. I think it requires too many levels of abstraction for me, but I think you can do it.
I'm sure.
Yeah. Some other things about the approach that we're implementing here is the modules are small. They try to do just one thing and to do it well. There's few strict rules to kind of encourage anyone to contribute, and that has been relatively successful. But we do make some very strong recommendations about the style, about these things that I mentioned, and the data formats to make sure that things are interoperable. There's, a few contributors. Some of them are also in the BioJS team. And maybe we can think of BioJS for visualization aspects and then BioNode for more of the handling. And I mean, we've been talking with Manny and with other the members of the two worlds have been talking quite a bit and have influenced each other quite a bit. And there's good opportunities to use these tools together as a combined ecosystem. Yesterday, somebody contacted us to make a hydro node initiative for hydroinformatics, whatever the heck that is. Anyhow, Bruno's the best guy for technical details regarding this. The Gitter is a good responsive place. I'll speed through the last points so we can go for drinks. You know how it's a pain in the butt to install software? Sometimes you have to do it yourself. Sometimes the sysadmins can do it, but it'll take forever. Well, Docker is an interesting way to package things, but Docker's a bit counterintuitive if you're a bit of a noob. This is a small wrapper for Docker. We call it oswitch because it switches operating system. And basically, here we type oswitch -l. It says, OK, these are the things that we've already used. Any Docker container works. Once you type oswitch and then one of them, it feels like you're in a different operating system if you have Docker installed. It's just a wrapper for Docker. And so you can just use the tools. Your computer then thinks it's a BioLinux here. So it's my MacBook thinks it's a BioLinux. All my files are in the same place. All my home directories are properly defined in the same way. Things just stay in the same place and just work. And then when I'm done with that, I can just go back. So you can go in interactive mode to enter BioLinux and then do a bunch of steps and then come out. Or let's say for a pipeline, you need several different pieces of software that are in different Docker containers. Well, you can use this in a single line. So here, in a single line, we just run BWA. It will create the Docker instance, go into it, do the config so that things are in the right place, run the software, and then get out again. That's one. Another one is that there's some challenge with gene predictions. It's the oldest bioinformatic challenge. We're still really bad at it. And we've made some tools to help visualize, when you have a gene set. Say you have a new gene prediction. You don't know how it is. Here, we just make a small plot that shows you, we blasted it. This is the length of your gene prediction. These are the lengths of all the similar things in the non-redundant database, or in UniProt. I don't know which one this was run against. Maybe this is biologically real, or maybe there's a problem with that sequence and it's artificially too long. There's a few other visualizations that can be useful to help prioritize how gene predictions should be fixed. And then finally, I'll just mention it here, we have an initiative for classroom use where we're recruiting many students to help curate gene predictions. So fixing a single bad gene prediction takes a few minutes, but when you have many species with many genes each, well, it becomes an impossible task, but with many people, even mountains can be brought down. I'll leave it at that. Thank you very much. I'm happy to take any questions.
Right, so I'll get started. So tell me more about your Java, your Indonesia trip.
Yeah, so when we spoke for the first time was in Singapore airport. And there, I guess that was last summer. I just spent 2 weeks in Borneo collecting a bunch of different ants. There I'm just going for diversity because, you know, as some people are starting to do 100,000 human genomes, some of us would like to do, you know, similarly large numbers of ant genomes. It's a bit more challenging to get the funding for 100,000 ant genomes than for the human ones. Yeah.
So in terms, how good are the genomes characterized with this? You obviously have many graphs on this table.
Yeah, so there's, I think, 15 published genomes. They're all crap. You know, they're all 5,000 scaffolds, 15,000 scaffolds. There's some linkage data, but, you know, then you lose 30% of the genome if you know, only consider the part that is in linkage groups. So there's quite a bit of potential there. For a lot of things, that's still sufficient. For others, it's problematic. So for kind of large-scale synteny analyses, it's problematic. And the trade-off is always, okay, do we invest more to fix these now, or do we wait 6 months for an amazing technology to do it for us? Yeah. So.
Any more questions?
I have a biological question,
Yeah.
actually. This region which is inverted in the little b and the big B,
Yeah.
how does this happen biologically? Is there, I mean, there is duplication, right? But what's the mechanism which evolutionary inverts and reinserts, and how often does it happen? And did you
I think it's
observe-
just a mistake that, you know, it happens regularly. I mean, when people try to kind of, the way that people did genetics a few years ago is you had some fruit flies or whatever, and then you would, you know, throw x-rays onto them until one of them was mutated, and different types of x-rays would lead to, you know, more double-stranded breaks than others. A double-stranded break, it can be fixed the wrong way around. And so I think that's, you know,
what happened in
that's
biology.
just what happened, yeah. Yeah.
The two different social structures within one species. You said it's happened with more than one species.
Yeah.
I think, but with different genes.
Different chromosomes.
Okay.
Yeah.
So I'm not a biologist, you might know. But still, it's interesting. This means that there must be some evolutionary advantage to having this feature.
In some situations, yeah.
Could you explain? Is there
Yeah.
a justification?
So we actually say that the two social forms, we think the two social forms are under balancing selection. So in some situations, it's more advantageous to be multiple queens, and in some situations, it's more advantageous to be a single queen. And that has to do with the queens from a single queen colony, they're able to get really fat, really big, and so they have large energy reserves. When they do their mating flight, they're able to fly at 300 meters altitude. That means that they can be carried by the winds very far. I think up to 120 kilometers or something like that. So they're really good at colonizing new habitats. So when a new habitat becomes available, they're there right away. In the multiple queen colonies, the queens stay much smaller, the young queens, and they do their mating flight at something like 3 meters altitude, and then they'll join an existing nest. So they have an advantage in a saturated habitat because if you have 100 queens in a colony together, all making 400 babies a day, they end up making a huge army, and they're able to outcompete any single queen colony. So in a saturated habitat, it's better to be multiple queen colony. In a If there's new habitats to colonize, it's better to be single queen.
For this to be advantageous, you don't want the two forms to diverge. They still need to interbreed, I would say.
So
But they seem hostile to one another somehow.
it's, I guess, just another form of genetic variation. So here in the system, the way it is, because little b individuals die, it seems that the multiple queen form cannot exist without the single queen form. Because in this, if you have a multiple queen colony, you will always retain the genetic material required to make a single queen colony. What will happen a million years from now, 10 million years from now, I don't know. So what we know is that in other species, you have some species where everything is always multiple queen, and some species where everything is always single queen. Maybe for single queenness to disappear from this species, somehow an additional genetic rearrangement would have to take place so that little b individuals, or whatever the subsequent thing is, are not, so that they don't die, so that the multiple queen colonies can exist on their own without having the genetic material for single queenness. Yeah.
So, really nice one. You said that males fertilize females once, and
Yeah.
then the sperm stays 30 years plus.
Until the queen dies, basically.
Is it the sperm or the diploid that stays?
No, it's the sperm. So the
How is that? How can a cell survive? So what is the balance?
I don't know. There's some really exciting molecular work to be done potentially. Looking at what are the molecules in the spermatheca, that allow them to keep the sperm alive for so long. Very little. So if you want to join and work on it, or find funds to work on it, there's some opportunities there, and you can think of good commercialization opportunities too. Yeah. Yeah.
The last software project I was understanding is that we have defined that in citizen science style, when you get them in the public, untrained, to help them
I think citizen science projects, to really work, you need something that people dream about. Things like the stargazing, or something that's really fun, and somehow people find protein folding improvement fun. Respect to the guys who made protein folding a game that people became passionate about. I think that's really challenging. So here, our approach is to use undergraduate students. There's multiple benefits. One thing is it means, we can suppose that the people have basic biological knowledge. Another thing is we can force them to do it. But a third thing, we hope to create engagement. So actually, when you're an undergrad, it's, you always do theoretical things. Here, we're giving them an opportunity to contribute, to look at real data, and to actually, do curations that will be used by real scientists. And so you can think of it for classwork, and that can also, be used as a teaching tool to teach them about how bad bioinformatics algorithms are, and teach them about gene structure and et cetera. But also, we can develop a mechanism for additional qualifications. So say, for the course, you have to do 50 things. Some of them are controlled exercises, some of them are random. We have students do them redundantly so that we can get, kind of use the wisdom of the crowd to weed out the crappy ones. It's easy to think of an incentive system whereby students might want to continue contributing after that specific episode of coursework. And kind of the really simple thing is to, if they've done 100 curations that have been accepted because the result was identical to other people or whatever, they do 100 of those, we auto-generate a PDF certificate thing, and then that's a line they can add to their CV. You can think of several things like that. Monetization works too. Yeah.
So when you did the ambiguous comparison, you
Yeah.
figured out there was one of them which was different. But actually, in Slack, there was another element which wasn't different. There was like when you go back in slides, like two which were marked red, G9, or
Yeah. Way at the beginning, you mean?
Yeah. So when you compare to B and so on.
Do you mean this one? Next. You mean this one?
Yeah, exactly.
Yeah.
That's EGM3.
Yeah.
What's called that?
So initially, this was actually the first one they found. And that one, well, once they found this one, all the variation of that one was eliminated. So kind of if you do an analysis with both together, it turns out everything you see here disappears if you take into account the effect of this one. And so this one is likely on the normally recombining part of the social chromosome. So there's some linkage, but it's not complete linkage to scleroderma.
Okay, so thank you very much.
Thank you.